This method can help answer key questions in the obesity field, such as identifying key factors and mechanisms that prevent a robust adipogenic response during adipose tissue remodeling. The main advantage of this technique is that it allows isolation of CD34 positive, CD31 positive endothelial cells from human adipose tissue, starting with small amounts of tissue.
We first had the idea for this method when we wanted to identify cells in human adipose vasculature that respond to adipogenic stimulation.
Visual demonstration of this method is critical, as the cell isolation steps are difficult to practice due to the oftentimes limited access to fresh human adipose tissue.
Collect human, omental, and subcutaneous adipose tissue from human subjects undergoing bariatric surgery. Immediately after tissue extraction, keep the human omental and subcutaneous adipose tissue in separate vials containing Hank's buffered salt solution with 50 micrograms per milliliter of penicillin streptomycin at room temperature. Once the sample arrives at the lab after the tissue extraction, carefully clean and remove fibrotic and cauterized sections of the tissue by using 70% ethanol swabbed scissors and tweezers. Weigh the adipose tissue and partition it into 5-gram aliquots for the collagenase digestion. Use 2 pairs of scissors to finely mince 5 grams of the adipose tissue in 5 milliliters of a collagenase solution in a scintillation vial at room temperature. Add an additional 10 milliliters of collagenase solution to the minced tissue for 15 milliliters.